Alan Gray here with the Microbiome First Summit, underwritten by the World Asthma Foundation. We're pleased to introduce our next speaker, Dr. Benoit Chasson. Dr. Chasson is Associate Professor at the French National Institute of Health and Medical Research in Paris, France. Dr. Chasson's new clinical research indicates that a widely used food additive, carboxymethylcellulose, alters the intestinal environment of healthy people, perturbing levels of beneficial bacteria and nutrients. These findings demonstrate the need for further study of this long-term impacts of this food additive on health. Today, Dr. Chasson presents his session, Ubiquitous Food Additive and Microbiota and Intestinal Environment. Dr. Chasson, over to you.
Okay, good morning everyone. So my name is Benoit Chasson and I am a research director and team leader in INSERM Paris in France. And today I'm going to talk a little bit about some modern stressors of the intestinal microbiota. So just to very briefly introduce my presentation, of course, I'm going to be very brief on the intestinal microbiota, which is, again, the complex community of microorganisms that's colonizing our gastrointestinal tract, and that's playing a lot of beneficial roles, such as protection against pathogenic bacteria, as well as maturation of our immune system. But what we know also is that if this intestinal microbiota is not kept in check and under control by the host, it can also drive some detrimental impact, such as the promotion of chronic intestinal inflammation and metabolic deregulation. So what we are studying in the laboratory is really trying to understand what's making this dual aspect of the microbiota and how a microbiota with beneficial impact for the host can actually turn out to become detrimental for the host. And the work that we have done in the last 10 years have demonstrated that what's called microbiota encroachment, meaning the ability of select microbiota member to penetrate the normally sterile mucus layer, which is represented here with the red bacteria that we named invaders. And we demonstrated that this microbiota encroachment and the presence of those invaders is actually key to drive the detrimental impact of the microbiota and move away from its normal beneficial interaction with the host. So this is another way of looking at it, where you can see on the left representation, you can see that the microbiota, which is labeled in red, is really kept at a safe distance from the surface of the epithelium, with this no man's land, no bacterial land at the surface of the epithelium. While on the right picture here in this presentation, you can see some bacteria that have penetrated the normally sterile mucus layer that get much closer to the surface of the epithelium. And this is exactly this phenomenon that's called microbiota encroachment. And again, what we have demonstrated during more than 10 years now is that microbiota encroachment that's demonstrated here is really important in driving chronic intestinal inflammation. So of course, the next very important question is what's driving microbiota encroachment in a way that can drive intestinal inflammation. And the example I wanted to give to you today relates to the use of various food additives, which are named dietary emulsifiers, and that are very, very highly used by the food industry in order to improve texture and extend shelf life. And this is linked to the fact that those food additives are actually stabilizing emulsion. So every time you have a packaged product that's containing some fat component, so I mean chocolate bar, ice cream, salad dressing, all those products will be loaded with emulsifiers to stabilize the emulsion. And what we did in our initial study that was published 7 years ago now was to use two of them. So the first one being carboxymethylcellulose, which I will refer to as CMC in this presentation. And we also used polysorbate 80, which I'm going to refer to as P80. And our first approach was to treat various strains of mice with either CMC or P80 for up to 3 months. And what we first analyzed, again, relates to microbiota localization. So we have a way to keep this mucus layer completely intact, and after using confocal microscopy, we can determine what's the distance that separates the microbiota in red from the surface of the epithelium. So basically, we are able to determine the thickness of the no man's land that's present at the surface of the epithelium. And as you can see here, in control untreated animals, we can see that microbiota is effectively kept at distance, with a distance that's separating the microbiota from the surface of the epithelium of approximately 25 to 30 micron. While when we treated mice with either CMC or PAT, you can see that now we have what's called microbiota encroachment. You can clearly see that the mucus layer is colonized by various members of the microbiota. You can see that even sometimes you can see here on the bottom right part of this PAT picture, you can see one bacterium that's directly adhering to the surface of the epithelium. And this is something obviously that you want to be protected against, and that can be really, really detrimental for the host. In terms of quantification, this is represented here on the histogram. You can see that just by treating either with CMC or PAT, we had a decrease of the distance that separates the microbiota from the epithelium by approximately twofold. So again, demonstrating that just consuming dietary emulsifiers is definitely sufficient to promote pretty strong microbiota encroachment. What we like to study in the laboratory also relate to microbiota function, not only its localization, but also its ability to promote and to activate innate immune signaling. So for this, we like to quantify the flagellin on the left and lipopolysaccharide on the right that are expressed by microbiota, and we know that those molecules, which again can be expressed by the microbiota, are really, really potent in their ability to activate immune signaling. And this is linked to activation of receptors like TLR4, TLR5, as well as NLRC4. And what you can see is in water-treated mice, the fecal level of flagellin on the left and LPS on the right is staying pretty stable over time and relatively low compared to CMC and PAT-treated animals in orange and purple again, where you can see that according to along the treatment, you can see that the fecal microbiota is starting to express much more fecal level of flagellin and fecal level of lipopolysaccharide. So meaning that not only the microbiota localization is impacted by consuming food additives, dietary emulsifiers, but also that the microbiota is starting to express much more pro-inflammatory molecules such as flagellin and LPS. So obviously this was not without any detrimental consequences for the host, and we were able to demonstrate, for example, using IL-10 deficient animals. You can see that in those animals in blue that were left untreated, those mice are genetically susceptible to develop colitis, and it's indeed the case. You can see that after 3 months of treatment, approximately 40 to 50% of them developed colitis. But what's pretty striking here is that if we treat those mice with either CMC or PAT during this period of time, you can see that now we have a colitis incidence of up to 80 to 85%. So there is definitely much more animals that develop colitis compared to the control untreated group. And not only there is much more animal developing colitis, but what's not presented here in this presentation is that the colitis that's developed by those animals are much more severe. So we have some marker, some quantitative marker of histology, molecular, as well as cellular inflammatory marker in those animals, demonstrating that indeed the colitis that's observed in CMC and PAT treated mice is much more severe compared to the colitis observed in control untreated mice. So this was again using animals that are genetically susceptible to develop colitis and clearly demonstrating that just consuming chronically dietary emulsifier is sufficient to promote colitis incidence as well as colitis severity. And what we did next was to use wild-type host, so without any genetical predisposition to develop any type of colitis. And what you can clearly observe is that it was also sufficient to increase some parameter of intestinal inflammation. So for example, here we use the very sensitive marker of intestinal inflammation, lipocalin-2. And you can see that after 2 months of treatment, this marker is definitively increased compared to the animals that were left untreated in blue. And clearly demonstrating that even in wild-type animals without any genetical susceptibility, consuming CMC or PAT is sufficient to drive chronic intestinal inflammation in those animals. So of course, this was again not without any consequences, and having such inflammatory state of the intestine for such a long period of time was having a lot of detrimental consequences, and notably in terms of metabolism. And you can see that when we evaluated, when we looked at body weight over time in those animals, you can see that the one that were treated with CMC or PAT gained more weight compared to the water untreated animals. And this is pretty clearly evident here on the left curve, where you can clearly see that after 2 months of treatment, the CMC and PAT treated animals start to gain more weight compared to the control group. And this was also evidenced by a glucose tolerance test, where you can clearly see that the CMC and PAT treated animals develop some inability to properly manage glucose challenge in the glucose tolerance test. So yeah, not only dietary emulsifiers, the consumption of dietary emulsifiers drive chronic colitis in genetically susceptible hosts, but it can also drive chronic intestinal inflammation and metabolic deregulation in animals that are genetically unenhanced. So just to summarize what we did, and this was again published quite some time ago now, is that using conventional mice that you treat with dietary emulsifiers, this is sufficient to alter the microbiota composition as well as function in a way that drive intestinal inflammation. And we did we did some follow-up mechanism where we used some germ-free mice, so mice without any microbiota, and what we were able to demonstrate is that those animals are actually completely protected against the detrimental impact of emulsifiers. So demonstrating that if you remove the microbiota from the equation, you just simply remove the detrimental impact of dietary emulsifier. On the other hand, when we use in vitro microbiota system, and this was thanks to a collaboration with Professor Tom van de Wiel at the University of Ghent, what we observed is by directly treating an in vitro microbiota without any host components with dietary emulsifiers, this was sufficient to alter microbiota composition as well as function by increasing its pro-inflammatory potential. And this was very similar to what we observed by treating directly the animals with those compounds. And what's very interesting with this in vitro microbiota system is now we can collect those microbiota that were treated with dietary emulsifiers, transplant them to germ-free animals. And what we observed is that those animals were actually developing intestinal inflammation even if they never consumed dietary emulsifiers. They just received an in vitro treated microbiota that was treated with emulsifiers, but they never directly consumed those food additives. So clearly demonstrating that not only the microbiota is a direct target, but it's both necessary as well as sufficient to drive the detrimental impact of dietary emulsifiers. And this is acting as presented here through, again, direct effect on the microbiota that's going to drive its ability to penetrate the normally sterile mucus layer, get in close contact with the epithelium, express much more pro-inflammatory molecules in a way that's going to drive chronic intestinal inflammation as well as colitis in genetically susceptible hosts and metabolic deregulation. And what's really, really interesting with this model is that, again, the microbiota is the direct target of dietary emulsifiers, but not all the microbiota. So what we were able to demonstrate in some follow-up studies is that if you take animals with a microbiota of minimal complexity, so which is altered Schindler-Flora mice, so those mice are colonized with a microbiota of a minimal complexity composed of only 8 bacteria. And you can clearly see that if you treat those mice with a minimal microbiota, you do not recapitulate any of the detrimental effects of dietary emulsifiers. So when we look at inflammation using the inflammatory marker lipocalin-2, when we look at microbiota pro-inflammatory potential, looking at LPS and flagellin, as well as when we looked at microbiota localization on the bottom part of the presentation, you can clearly see that we did not observe any effect between ISF mice treated with nothing, treated with CMC, or treated with PAT. So clearly, this was highly reproducible, clearly demonstrating that those mice are completely protected against the detrimental impact of dietary emulsifiers. So this is quite interesting because this is driving the concept that you can have some resistant microbiota, such as the ISF mice, that are harboring a specific microbiota that's going to be completely protected against the detrimental impact of dietary emulsifier. On the other hand, you can have some susceptible microbiota that's going to host some specific species, such as the one present in normal conventional mice, that are going to detect and respond to dietary emulsifiers in a way that, so those bacteria will next be able to penetrate the normal visceral mucus layer and induce chronic intestinal inflammation and metabolic deregulation. So clearly suggesting that maybe there is an infectious aspect in the susceptibility to dietary emulsifiers. So of course we did some follow-up study on those aspects, clearly demonstrating that this is indeed the case, and that we can actually convert a resistant microbiota to a susceptible microbiota just by adding some new microbiota member in the equation that are going to detect the dietary emulsifiers. So there is definitely some microbiota member that are able to detect and to promote inflammation in the presence of emulsifiers, while some of the microbiota member, and I should say most of the microbiota member, are going to be protected against those compounds. So of course, the next important question relates to other emulsifiers, because everything I presented so far relates to CMC and PAT, which are again highly used, but they are definitely not the only one, and there is up to 60 that are approved to be used. So yeah, we definitely wanted to have a little bit of insight regarding other dietary emulsifiers. And for this, what we set up in the laboratory is an in vitro, our own in vitro microbiota system, which is named mini bioreactor arrays, and that was originally developed by Robert Britton as well as Jennifer Hartung. And this is a really elegant system, because what's allowing us to do is to cultivate up to 50 microbiota in parallel. So you can see in every single of those tubes here, you have an independent microbiota that's completely independent from all the other. And we can keep this microbiota, inoculate this system with various donors, and keep the microbiota stable for up to 3 weeks, and study the impact of various compounds in a high throughput manner. And this is exactly what Sabrine Naimi, one of my postdocs, did in the laboratory, by treating the system with various dietary emulsifiers, just to see what would be the detrimental or beneficial consequences in terms of microbiota composition and function. So first, what she observed was when she used CMC and PAT, this is represented here again in orange and purple, you can see that we were able to reproduce what we observed before. So here, this is an arrow under the curve, but just to be clear and to be brief, the higher the histogram is going, the more altered the microbiota was in terms of both composition and function. And you can clearly see that indeed, both CMC and PAT, when we treat in vitro microbiota system, is sufficient to alter microbiota composition and microbiota function. The pretty good news that was brought by Sabrine's work on those food additives, and that was recently published, is that there is actually few dietary emulsifiers that seems to be completely inoffensive in terms of their impact on the microbiota. And you see that this is, for example, the case for mono and diglyceride, which are compounds that are really, really highly used. And the fact that they do not have any detrimental impact on the microbiota is kind of a good news, because it seems that there is still some dietary emulsifiers that can be used definitively by the food industry. The not so good news was that most of the compounds that we tested were having some level of detrimental impact in terms of both composition as well as function on the intestinal microbiota. So clearly demonstrating that CMC and PAT are not the only one, and that this is actually something that's shared by most of the dietary emulsifiers used by the food industry. And the pretty bad news relate to the carrageenan, which are represented here in red, as well as some gum, so guar gum and locust bean gum, which appear to be highly detrimental for their impact on the intestinal microbiome. And this is quite interesting because there is some doubt and there is some data suggesting that carrageenan are actually able to promote chronic intestinal inflammation. And what we are demonstrating here, or at least suggesting, is that this is very likely driven by a direct detrimental, strong detrimental impact on the intestinal microbiota. So overall, it seems that most of dietary emulsifiers seems to be pretty bad for the impact on the intestinal microbiota in a way that can very likely drive chronic intestinal inflammation and metabolic deregulation. But there is some hope with the identification of select food additives, select dietary emulsifiers that seems to be highly without any detrimental impact on the microbiota. So of course, what's the human relevance of everything I presented so far, and this is something I wanted to bring to your attention. And of course, this was asked at multiple times by food agency as well as colleagues in the field. So this is why we decided to set up the first clinical trial investigating the impact of dietary emulsifiers in humans. And this study was named FRESH, for Functional Research on Emulsifiers in Humans. And this was an inpatient study. So patients were locked in an hospital room for the entire length of the experiment, of the clinical trial. And this was obviously made possible through an outstanding collaboration with Gary Wu and Jim Lewis from University of Pennsylvania, as well as Andrew Gewertz from Georgia State University. And what we did is represented here. So we had participants coming at the hospital at day 0. And for 3 days, the first ones, everybody went on an additive-free diet. So the diet that we were producing to them and giving them, that we know it was completely free of any type of additives, and especially, of course, dietary emulsifiers. And after about 3 days of washout, we had an oral glucose tolerance test performed, as well as we collected biopsies. And then half of the participants stayed on this same additive-free diet, while the other half of the participants were treated with carboxymethyl cellulose for 11 days. And so we focused only, so there is definitely a lot of limitation in this study. First, we focused only on CMC, because it was already pretty ambitious and we wanted to have something that was feasible. So we focused only on CMC. You can see it's for a pretty relatively short period of time, only 11 days. And also, more importantly, it was really low in terms of number of participants, because we only had 16 participants total. So it was really more to generate new hypotheses, and it was definitely a pilot trial, just to try to start to investigate if what we observed in a preclinical and in vitro setting before will hold true in human. And of course, throughout the study, we had some stools that were collected on those participants. So first, what you can observe here is at the end of the dietary supplementation with CMC, we were able to reproduce the detrimental impact the impact on microbiota composition, where you can clearly see that all the untreated participants in blue were not impacted by the diet, while all the participants in orange that were consuming CMC are clustering apart and were having some alteration in their microbiota composition. So we observed that microbiota composition on the untreated participants were relatively stable, while microbiota composition and especially beta diversity as well as alpha diversity was impacted in participants that was treated with CMC. So really encouraging, because we can alter microbiota composition even on such a short period of time and only focusing on one dietary emulsifier. So this is further exemplified here, where you can clearly see that in control participant, the alpha diversity, which is again the richness, the diversity of the microbiota is staying pretty stable over time, while in the participants that were treated with CMC, you can see this relatively small, but nonetheless functionally important drop in alpha diversity after the consumption of CMC. And again, this was observed even if we used only one dietary emulsifier, on a very relatively short period of time. So one can imagine what would be the consequences of eating food that's containing multiple dietary emulsifiers in combination that can very likely have synergistic effect for an entire life. You can definitely start to imagine what can be the consequences in terms of microbiota richness as well as microbiota composition. What we did also was relating to microbiota function, and for this we did some metabolomic analysis, and this was thanks to a collaboration with Andrew Patterson here as well that I wanted to highlight, and we were able to follow various fecal metabolites of importance in the fecal samples collected from those participants. So here, this is only the control patients throughout time. So you can clearly see that there is some variation, and not all the metabolites are present at the same quantity, but you can see that it's pretty stable throughout the study, and there is not a clear impact of the diet per se. However, what was pretty striking relates to the use of the supplementation with CMC, where you can clearly see that during the CMC supplementation period, so between day 6 and day 14, so as soon as the CMC was reaching the gastrointestinal tract, you can see that we observed a pretty drastic depletion in numerous fecal metabolites. And this was true for some bile acid, and this was true for amino acid, but even more importantly for some short-chain fatty acid. So yeah, we know the potential beneficial role played by short-chain fatty acid in the gastrointestinal tract. So having one food additives that can so efficiently and so rapidly deplete the level of short-chain fatty acid within the gastrointestinal tract is pretty important finding. And here again, even if we focus only on one dietary emulsifiers for a relatively short period of time. And next, what we did also was to look at microbiota localization, and it was really interesting finding. So here again, more generating hypotheses than giving some clear definitive answer. But what we observed is actually in the CMC treated participants, we observed two subgroups of participants, where you can clearly see that most of the participants were not impacted by consuming CMC. So even if they had some alteration in microbiota composition and function, microbiota encroachment was not impacted by CMC. While for two of the participants, we observed a very, very strong microbiota encroachment, even after only 11 days of exposure. And what was really, really interesting is that those two participants that were presenting and having microbiota encroachment after consuming CMC were also the two participants with the most severe impact on microbiota composition, microbiota richness, and fecal metabolites. So it seems that there is definitely here again this concept of CMC insensitive and CMC sensitive participants, where you can have some participants that will have microbiota that will be less sensitive to dietary emulsifier CMC, while it seems that some participants are harboring a microbiota that makes them inherently much more susceptible to develop the chronic intestinal inflammation and potentially metabolic deregulation after consuming dietary emulsifiers CMC. So this is why we decided to went back to mice after having those data on human. We decided to went back to mice to try to test this hypothesis and to try to understand if really the microbiota is actually driving the phenotypical response to CMC exposure. So what we did was to use germ-free IL-10 animals. So again, mice without any microbiota, but mice that are genetically susceptible to develop colitis. And we took 4 groups of mice, and we colonized those 4 groups with 4 individual microbiota that were coming from the clinical study. So for this, we selected 2 microbiota from participants that were exposed to CMC, but that seems to be pretty insensitive to dietary emulsifier CMC, and especially in case of regarding microbiota encroachment. And we also took 2 microbiota from the 2 clinical study participants that were the most affected by the presence of CMC. So this is the points that are represented here. So you can clearly see 2 participants that seems to be CMC insensitive, and 2 participants that seems to be CMC highly sensitive. And what we did was to colonize those mice with those 4 individual microbiota, and then we splitted each group in 2 by treating half of the group with water and half of the group with CMC. So just to try to understand what would be the impact of just transferring the microbiota to animals and to see if the microbiota is actually holding the CMC sensitivity or not. So what we observe in terms of colon length, which is a good proxy for intestinal inflammation, as well as histological score, is pretty clear here for those groups colonized with insensitive microbiota. So you can see that those 2 first graphs, those 2 bars, first bar, sorry, are of mice that were colonized with microbiota from an insensitive donor, treated either with water for the first bar or CMC for the second bar. And you have exactly the same data here after for the other insensitive donor. And you can clearly see that when we colonize mice with microbiota from those insensitive donors, they are not developing chronic intestinal inflammation, even if we treat them with CMC. So it seems that those microbiota are actually pretty healthy. Those mice are not developing chronic colitis, even if this is a mouse model of chronic colitis. And even if we add CMC into the equation, it seems that CMC is not sufficient to drive colitis in mice colonized with those microbiota. But now look at what we observe when we colonize mice with sensitive microbiota. So you can see you have the 2 red here are mice colonized with 1 microbiota from 1 sensitive donor, and the 2 pinky bar are mice colonized with microbiota from another sensitive donor. And what's clearly evident here from both looking at the colon length as well as histological score is that those microbiota are not sufficient per se to drive chronic colitis. But as soon as we add CMC in the equation, you can see that colon length is decreasing and histological score is having a pretty dramatic increase, which demonstrates here that indeed there is something inherent in those microbiota that can drive the susceptibility to dietary emulsifiers consumption. And having CMC only seems to be not sufficient. So having those food additives is not sufficient. Having this specific microbiota is not sufficient, but the combination of both is sufficient to drive chronic colitis. So I hope you understand what can be the consequence in terms of patient stratification and IBD patient healthcare management in terms of these findings regarding the impact of the alimentation and the consumption of various food additives that they can have in their diet. And we did also some analysis on microbiota localization, and just very briefly, what you can clearly see is in mice colonized with insensitive microbiota, consuming CMC is sufficient to promote some level of microbiota encroachment. I don't think this was significant, but when we did exactly the same with mice colonized with sensitive microbiota, you can see that microbiota localization at steady state, so under water condition, is relatively stable between those 4 donors. But when we add CMC again in the equation, you can see that we are able to reproduce what we observed in the clinic with a pretty strong microbiota encroachment in those two participants. So it's really very interesting to us, and this is a study that we are trying to publish now, is that the microbiota is definitively holding the susceptibility to dietary emulsifiers, and maybe we can use this microbiota to predict who is going to be susceptible to dietary emulsifiers and who is going to be protected against the susceptibility to dietary emulsifiers. So of course, this is something that we are actively doing in the laboratory, and for this, actually, what we are doing is we are going back to our in vitro microbiota system, because this in vitro microbiota system is very good to be able to screen multiple donors and to try to see how many microbiota are susceptible and how many microbiota are resistant to dietary emulsifiers. So you can clearly see that this is something that we can do in the laboratory, where you can clearly see that there is some microbiota. And again, we have some very good correlation with our clinical study here. I'm not going into much detail here, but you can clearly see that there is some microbiota, roughly 80% of them that seems to be fully protected and fully insensitive to CMC exposure. You see that their microbiota composition is staying pretty stable over time, while there is some microbiota which are represented here in red, that when we put CMC in the equation, have a pretty drastic impact in terms of microbiota composition, but also in terms of microbiota function, so expression of pro-inflammatory potential. So it's really elegant to us because we can now use this in vitro microbiota system to screen a lot of donor and see if we can do some correlation between what we observe in the clinic and what we observe in preclinical animal model. And what's, of course, what the big question is what the molecular signature of such a sensitive microbiota. This is something we are actively working on. But basically, when we look at taxonomical data, so basically, who is present in the microbiota composition, so who is present in the community, which species are present, we are not able to do any type of prediction in terms of microbiota that's susceptible or microbiota that's resistant. However, when we analyze metagenomic data in terms of functional, so when we look at genes and bacterial DNA that's present, so the whole bacterial DNA, not only who is present, but what the bacteria are actually expressing within the gastrointestinal tract, we are able to identify some markers that perfectly correlate with susceptibility. So you can see here in blue are donors that are highly protected against CMC. In red, you have the relative abundance of some metagenomic marker that were observed in microbiota that we know are highly susceptible to CMC. So we have everything in hand now to be able, using metagenomic data, to predict what's a resistant microbiota and what's a susceptible microbiota. So I hope you can see what can be the application of such data where, for example, for the IBD community, we can perform some metagenomic analysis, which is getting cheaper and faster and cheaper in the laboratory. And using those metagenomic data, we can actually predict if these participants can benefit from stopping dietary emulsifiers, or if you can keep hitting dietary emulsifiers without any detrimental consequences in terms of chronic intestinal inflammation and disease activity throughout time. So this is the conclusion slides, and this is where we are currently, where dietary emulsifiers is definitively a bad food additives, and especially CMC, PAT, and carrageenan that can drive microbiota encroachment and promote chronic intestinal inflammation. But this is true only in select microbiota that are holding some specific bacterial species that's making the participants and the animals CMC sensitive, while you can have some microbiota that seems to be highly protected against the detrimental impact of dietary emulsifiers. So of course, I wanted to finish by thanking everybody in the laboratory that working on those aspects in Paris, in my team here in Paris, and I'm really lucky to have a beautiful team of researchers that's helping me on a daily basis. And of course, I hope you realize that this is a highly collaborative project. So I presented you a lot of data that were generated thanks to a lot of various collaboration throughout the world, and everything was made possible thanks to those collaborations, as well as thanks to our funders that I wanted to acknowledge here on the bottom of my slides. And with that, I wanted to thank the organizers for giving me the opportunity to present all of these data. I hope you enjoyed it, and I would be really happy to answer your question following this virtual session. So yeah, thanks again for the invitation, and thanks again for your listening and for your attention. Thank you.